Hello, today we welcome Professor Quinn to this interview. Thank you very much for joining us. So Professor Quinn, please tell us a little bit about yourself.
Okay, yes, my name is Joaquim Segalés, but everybody calls me Kim. I'm a veterinarian, and I also nowadays full professor at the Autonomous University of Barcelona in Spain. However, I am also adscribed as a researcher at the Centre de Recerca en Sanitat Animal, which stands for Research Center for Animal Health, which is a governmental institute belonging to the Catalan government in Spain. In general terms, I have been involved in the research of swine infectious diseases almost for 30 years. And since 2014, I'm working with zoonotic coronaviruses. We started working with MERS coronavirus in 2014. And of course, in the last 2 years, we have been working quite a lot on SARS coronavirus too. In fact, my specialization is basically on veterinary pathology as well as swine medicine.
Nice. So could you please tell us a little bit more background on your research topic?
Well, in fact, as I told you, my real topic has always been the swine infectious diseases, because I have been working with pigs for a long period of time. And since 1997, we were involved in porcine circovirus too, one of the most important viruses in the swine industry, because especially in the late 90s, early 2000s, caused lots of problems to the worldwide industry. Therefore, and this was the topic of one recent review paper published at Veterinary Sciences, in which we tried to update all the issues related with the PCV2 diagnosis. Because we know that the diagnosis of the disease has been more or less the same for a long period of time, but it's clear that it has to be adapted to the current situations. But anyway, since 1997, with this particular viral disease, we have been working with many research projects, contracts with different companies, and especially to learn more about pathogenesis, epidemiology, immunity, diagnosis, especially as I indicated before, but also control and prevention. And very importantly, the last 10 years, it has been focused, I wouldn't say exclusively, but mainly on diagnosis and prevention by means of vaccination.
Very nice. So next question is, what really makes you want to research this subject? Did you get any influence from other people?
Well, this is a very interesting question. In fact, everything started really in 1997 in this particular subject on porcine circovirus 2, because I attended a meeting in Canada, and it was presented one new disease named post-winning multisystemic wasting syndrome, with the acronym PMWS. Just after the meeting, my perception was I've never seen this condition in Spain. But it was amazing, because 2 months after this meeting, we diagnosed the first case of the so-called PMWS in Spain. And this teach you that you are not able to diagnose what you do not really know. And this is something that marked a lot of my life research during the last 25 years almost. Because at that time, we observed that it was a terrific disease, PCV2 systemic disease, what it's nowadays known as the former PMWS, caused lots of mortality. And it was a really devastating disease for many farmers worldwide. And of course, since we were working on diagnosis, the novel disease was a real challenge. So what we pretend at the very end, it was to try to know more about the disease and try to help as much as possible to the farmers and the veterinarians and the swine industry as a whole. Of course, 25 years of particular research on one topic means that the interactions with other people has been a constant issue. And we have been participating in different European projects, in different contracts, etc. So this means that the number of scientists working on PCV2 worldwide is rather important, but I consider most of them as a big family, because we have been working together for a long period of time already.
I see. It must be really exciting for you to discovering something new, and that's why you want to study in this subject. So our next question is, what is the interesting or innovative part about your research? Are there any limitations when you are doing your research?
Well, at the very beginning, besides the diagnostic work, because of course it was very, very important to set the criteria for diagnosing the disease, the most important question was how this disease works, which are the pathogenic mechanisms of the disease. And for a period of time, I would say before the advent of vaccines, and this means almost 10 years, between 1997 and 2007, our major focus was how to understand the disease, which were the triggering factors, why a particular virus like PCV2, which is worldwide distributed, caused disease in only some farms. And this is one of the interesting issues that still in force, because nowadays we are dealing with viruses, bacteria, which are highly widespread, but we do not really know why in some cases there's disease and not in some others. However, it's clear that when by the years 2006, 2007, the vaccines came into the worldwide market, well, they represented a terrific revolution for the swine production because they were very, very, very effective. In fact, I would say that probably in the last 40 years, PCV2 vaccines have been the most effective products that reach the market. And what happened? As long as you have a good solution for a problem, the need for basic research is decreasing. And then it's very, very difficult to get money to invest in basic research. And there are still a number of question marks for which we do not have answer.
Wow, it sounds like you saved the entire animal kingdom. So our next question is, what are the challenges that we are facing in the current situation? Is there any difficulty you have experienced regarding your research?
Well, as I told you, PCV2 vaccines are extremely efficient. And this has not changed over the time in the sense that, well, the virus has been evolving. It's true that what we call genotypes, let's say the different types of viruses or the variants of those viruses, of course, have changed. And nowadays, 2022, we recognize up to 9 different genotypes. However, apparently, all genotypes are cross-reacting among themselves. What does it mean? This means that infection with one particular genotype protects against infection with another genotype. Therefore, even we have a number of genotypes, it looks like that there's only one serotype. And this means that vaccines that, at least in some parts of the world, that are based specifically on genotype A, PCV2A, are able to protect. Of course, it could not be full protection, but a wide protection against the rest of genotypes. And this is good news, because this means that even initial vaccines still work and work quite nicely. However, what has changed really during the last 10 years is that by mass vaccination, we are vaccinating almost all farms worldwide. We have changed the epidemiology of the virus. And this is what we try to highlight in the veterinary science paper recently published. Because in the new epidemiological scenario, PCV2 infections tends to be earlier on. And sometimes when we are vaccination or when we are applying the vaccine, which is mainly at weaning, this means 3-4 week old animals, might be we are reaching too late to get full protection. And therefore, this means that we have to reschedule how to vaccinate and get the most from the vaccines. Therefore, this is something that we must confess, diseases evolve. And what really works at a given time point might not work subsequently. But it's not a matter of vaccines losing efficacy. It's a matter of changing the epidemiology of the virus.
Cool. So it sounds to me that further research will be needed. So is there a next move for you? What are your main interests in your following research?
Well, one of the issues in which we have been focused during the last few years is the fact that since we have excellent vaccines, and we know that those vaccines protect against the disease, then the point is try to get the most from those vaccines and try to avoid those epidemiological situations by which early infections makes that vaccination comes too late. So from this point of view, I believe that there's still room for improvement in the sense that by means of monitoring the viral infection, knowing when there's a potential danger and the need of rescheduling vaccination is something that still applies. Of course, if you would ask me from a more basic science aspect, I would tell you that I would enjoy continue doing research in those pathogenic mechanisms because there are still a number of issues which are unknown or poorly known. However, as I told you before, to get money for those basic research is nowadays really, really very difficult.
I'm sure we all can appreciate that you have resolved 99% of the problems out there already. And we wish you the best luck for your ongoing research. So before we end, is there any acknowledgement you would want to make?
Well, certainly yes, because if you have been working 25 years with one single pathogen, for example, this means that lots and lots and lots of people cross alone. And especially, I'm talking about field veterinarians, farmers, that of course they experience the disease, they suffer from the disease in their herds, and it was quite frustrating sometimes. However, from the research point of view, there are a number of people that I would like to mention, such as Mariano Domingo, Maria Calsamiglia, Marina Sibila, Tuija Kekarainen, as well as many, many PhD students and postdoctoral fellows that they did a great, great, great job in terms of research on PCV2. In fact, nowadays, 2022, we still work with PCV2. And hopefully we will keep working with this virus the next few years as well. Thank you very much.
All right, Professor Quim, thank you very much for joining us, and we wish you and your team the best luck in the future.
It has been my pleasure. I really appreciate it, Ron.